My good friend George Stigma was walking down the street, and he thought to himself, what's the meaning of life? All I do is convert carbon dioxide into oxygen, all day, every day. He thought to himself, why was he created? Why was he here? Why was he alive? Would anyone care if he disappeared? Does the world really need plants? He sat down on a bench, drowning in his deep thoughts. Dennis Hopper, the frog, was out hopping as he does every Wednesday afternoon. He stopped as he saw George sitting on the bench next to the little pond. He hopped up to him and sat down beside him. "Oh, George, what is that that's bothering you this wonderful afternoon? Is it the bees again?" "No, my good friend, something deeper and more frustrating." "Well, what can be more frustrating than those silly bees?" Dennis said and patted George on the patter. "I can't understand the meaning of life. I feel useless, Dennis." "That is ridiculous. Without plants and flowers, we would all die. Let me explain." George began speaking about something called photosynthesis. The sun's rays hit the plant's pigments, which captures the energy and transfers it to the thylakoid. In the thylakoid, ATP and NADPH is created, and O2 is created by splitting water. ATP and NADPH are transferred to the Calvin cycle in the stroma. The energy from ATP and NADPH, and with the help of CO2, create high energy sugars. George nodded continuously as Dennis went on with cellular respiration. "Now I, as a frog, I breathe the oxygen you have created, and I use the high energy sugars in a process called cellular respiration. The process is split into 3 stages: glycolysis, Krebs cycle, and the electron transport chain. If we take glucose as an example of a high energy sugar, this is how it would occur. Glucose enters glycolysis and is broken down into 2 molecules of pyruvic acid, and ATP and NADPH is created. The 2 pyruvic acid molecules are now moved to the Krebs cycle." Dennis went on and on, but George listened and took notes as Dennis began speaking about the Krebs cycle. In the Krebs cycle, pyruvic acid is broken down into carbon dioxide in a series of energy extracting reactions. In the process of breaking down the pyruvic acid, a lot of NADH and FADH2 and ATP is created, and the NADH and FADH2 is sent to the electron transport chain. The carbon dioxide is released and ATP is stored. High energy electrons from NADH and FADH2 are passed from carrier to carrier down the electron transport chain. Water is formed when oxygen accepts the electrons in combination with hydrogen ions. Energy generated by the electron transport chain is used to move H+ ions across the inner mitochondrial membrane and into the inner membrane space. H ions pass back across the mitochondrial membrane through ATP synthase, causing the base of the synthase molecule to rotate. With each rotation driven by movement of H+ ions, ATP synthase generates ATP from ADP. All of this leaves me with a lot of ATP. We can now think, run, breathe, and a lot of other things, but without plants, nothing of this would be possible." George was amazed. Never had he ever heard about all this. "Tell me more, my good friend. Teach me about proteins." Dennis, surprised that George knew these names, was about to ask George how he knew all of this. But because he's a frog, he went on talking. Transcription happens in the nucleus. RNA polymerase uses a segment of the DNA as a template to make mRNA. The mRNA travels to the cytoplasm, where its genetic information is translated by ribosomes to code for proteins. To translate the code, the ribosomes read mRNA 3 bases at a time, which is called a codon. tRNA brings 1 amino acid to each codon. Then the amino acids link together to form proteins, which either are used in the cell or outside of the cell. George was now in tears. He finally understood his cause in life. How does the body make more of those amazing cells? Well, that is something called mitosis. Do you want to hear about it? Oh yes, please, tell me everything you know. In order for the body to grow and repair, the body needs to go through cell division. Cell division has different parts: interphase, mitosis, meiosis, and cytokinesis. This is how a cell divides. First, it's the G1 phase. The cell grows and synthesizes proteins. After that, the cell enters S phase, where the cell duplicates its DNA. The S phase is followed by G2 phase, where the cell just prepares for cell division. Now it's time for mitosis. Mitosis is divided into 4 phases. The phases are prophase, metaphase, anaphase, and telophase. In the first stage, which is prophase, the genetic material inside the cell condenses and the chromosomes become visible. During metaphase, the duplicated chromosomes line up in the center of the cell. Now the spindle fibers connect to the centromere of each chromosome. In anaphase, the sister chromatids separate and are pulled to different poles of the cell by the spindle fibers. Telophase is the last phase where the nuclear envelope reforms. Now mitosis is over, but the cell is not divided yet. Cytokinesis is when the cell splits into 2 daughter cells. Now the new cell grows and produces proteins before going through the whole phase again. Dennis had now ended his lecture for George. He hopped off the bench and started hopping again. Wait! How can I ever repay you, Dennis? Dennis stopped. He turned around. Just keep doing what you do, and everything will be fine.